Welcome to this edition of Clinical Clips, an accredited continuing education activity. This brief expert video will spotlight the daily hot topics from the conference.
Hello, I'm Laura Waters. I'm a consultant physician in HIV and sexual health at the Central and North West London NHS Trust in London, UK. And welcome to episode 2 of Clinical Clips from the HIV Glasgow 2022 conference. The first study I'll summarise was an analysis of resuppression rates in 4 randomised trials of dolutegravir, efavirenz, or protease inhibitor-based therapy, including more than 3,000 participants. The question they were addressing here is whether or not people were more likely to resuppress on an integrase-based regimen than an NNRTI or boosted PI. So they looked at 3 first-line studies. There was one advanced study or second-line study called ViiV. By combining data from all of these analyses, they looked at whether or not staying on the same regimen led to resuppression, and the rationale being that sometimes adherence counselling within a trial achieves resuppression without changing ART. Now, what they showed for all studies bar Dolphin, people were more likely to resuppress on a dolutegravir-based regimen than an NNRTI or boosted PI. In Dolphin, there was no difference, but as a study undertaken in pregnant women, there were quite high rates of treatment cessation after birth. The conclusion was that leaving people on a dolutegravir-based regimen can be a reasonable thing to do, and indeed the South Africa guidelines now recommend staying on dolutegravir despite non-suppression as long as no integrase resistance emerges. And I guess the caveat here is monitoring resistance at population level is going to be critical if this strategy of remaining on a failing regimen is to be supported. The next study I'll summarise, sticking to the theme of injectable cabotegravir rilpivirine, was a beautifully presented multivariable analysis of the predictors of confirmed virologic failure in the registrational trials for injectables. That's ATLAS, FLAIR, and ATLAS-2M. They've already shown that there were 3 key factors that predicted confirmed virologic failure at week 48. That's rilpivirine resistance at baseline, and that's on proviral DNA sequencing, having subtype A6 or 1 virus, and finally having a BMI greater than 30. This analysis looked at data out to year 3. What they showed was those same 3 factors were predictive, as were predicted trough levels of cabotegravir and rilpivirine. But if we focus on those 3 factors, people with none of those factors, overall, there was a 0.4% rate of confirmed virologic failure at year 3. Remembering this is a combination of 4 weekly and 8 weekly dosing. People who had any one factor, confirmed virologic failures at year 3 were 2%. However, if it was only having a BMI above 30, that was the only one factor someone had, that risk was 0.5%. And this is reflected in the fact that rilpivirine resistance, particularly an A6A1 subtype, are more predictive of virologic failure than BMI. People with 2 or more of those risk factors, it was approaching a 20% confirmed virologic failure rate at year 3. Now, for me, if possible, I'd like to see those different factors analysed looking only at 8 weekly dosing, because certainly in Europe, that's the only dose that's licensed. But what I think this analysis does do is help us counsel people a little more accurately. I don't think we can entirely exclude that someone might develop a detectable viral load despite on-time injections, but I think we can say if they have none of those 3 key risk factors, that risk is very low indeed. And analysing this type of post-hoc data to better implement precision medicine, I think, is a great move. And finally, 2 eagerly awaited abstracts looking at the islatravir safety signal that led to its development being paused at the end of 2021. Islatravir is an NRTTI, a nucleoside reverse transcriptase translocation inhibitor, showing great promise for HIV treatment and prevention, but an association with reduced total lymphocyte and CD4 counts led to the pausing of the studies while that safety signal was explored. Now, we've not heard much since the trials were paused, so it was really great to see this data. The first abstract was a modelling analysis looking at the correlation between drug exposure and declines in total lymphocyte and CD4 counts. They shared that islatravir triphosphate, because like other NRTIs, islatravir is phosphorylated intracellularly, that accumulation and high levels in cells leads to apoptosis. And their aim was to model what dose of islatravir would be safe, but also effective. So by looking at across the phase 2 and 3 treatment and prevention studies, they concluded that a 0.25 milligram daily dose, rather than the 0.75 milligram daily dose that had been selected for phase 3 studies, that lower dose would be efficacious against both wild-type virus and virus harboring the M184V or I mutation, and achieve similar total lymphocyte count and CD4 rises as standard non-islatravir containing ART. The second islatravir abstract was an elegant analysis of Protocol 011, which was a phase 2 trial looking at islatravir and doravirine dual therapy compared with doravirine, tenofovir, and lamivudine 3 drug therapy. So they showed data up to week 60, which demonstrated this dose exposure relationship. So people on the lower dose of doravirine, the 0.25 milligram, in this phase 2b study, they actually saw fairly similar total lymphocyte and CD4 counts as people on the standard triple therapy arm not containing islatravir, if anything numerically slightly higher, though the numbers are small. People on 0.75 and particularly 2.25 milligrams saw relative declines in those cell counts, and there was the same analysis undertaken for B cells as well. At week 60, everybody in the islatravir arms switched to a 0.75 dose because that was the original dose planned for phase 3. So you could actually look at the impact of switching dose. So people already on 0.75 stayed on 0.75 and their counts remained stable, showing there's no cumulative risk. People who are on 2.25 and went down to 0.75 saw an improvement in total lymphocyte and CD4 counts. And people who are on 0.25 and increased to 0.75 saw a decline. So a really quite elegant way of demonstrating this dose response when it comes to those hematological parameters. Now, importantly, there was no association with category C infection events, and there was no impact on hemoglobin, neutrophils, or platelets, and that means a 0.25 milligram islatravir daily dose is being taken forward. So whilst it feels like we've been waiting quite a long time for this data, I think it's really reassuring that by understanding this dose response and the model showing that there's predicted efficacy against wild type and 184 harboring virus, that there's still hope that islatravir will become an important part of our treatment and prevention choices. Thank you so much for joining us for the second and final episode of HIV Drug Therapy Glasgow 2022 Clinical Clips, and please visit the landing page to view the slides and previous clips from this conference.
We hope you found this activity useful and educational. To receive continuing education credit and to download your printable certificate, please return to the activity webpage and click on the Claim Credit button associated with today's clinical clip.